Dr. Nabil Naggar. His talk will be Beyond the Usual.
اللجنة
Track Beyond
المنظمة
the Usual.
للمؤتمر، وعلى رأسها الأستاذ الدكتور طلعت عبد العاطي، وأيضا الأستاذ الدكتور يحيى غانم، على ما يقدمونه من دعم وإصرار على نجاح هذا المؤتمر السنوي في كل عام. اليوم الحقيقة أنا هتكلم عن شيء جديد شوية، ولكنه بعض الناس مش عارفه جديد قوي. بنتكلم عن الريج أو الريسبتور أوف ايج which stands for the advanced glycation end products. يمكن كلنا نعرف as working in the diabetes field what is the meaning of age and what is the rage. إنما إحنا هنتكلم عن ال unusual about this subject. These are my credentials. And as an introduction, we all know that the ages or the advanced glycation end products are the products that are resulting from an enzymatic glycation products of the aldehyde group, which is present in reducing sugars, and the free amino groups of the macromolecular substances, زي proteins or lipids and other nucleic acids in the body. Under the condition of hyperglycemia, there is an increased production of the advanced glycation and the products, and this process is known as the Browning reactions or Miller reactions. And of course, as these products are present, there are receptors to receive it and send signals to other tissues. And these are the receptors for ages, which is the rages. احنا كنا فاكرين زمان انه الادفانسد glycation in the products one receptor that is present in different tissues. However, it has been known that the age receptors are not only one receptor, one type of receptor, but there are a lot of receptors. RAGE 1, RAGE 2, RAGE 3, and other receptors that are age-binding proteins, such as the scavenger receptors. These receptors are members of the immunoglobulin superfamily, and it is mainly responsible for signal transduction and legal induced upregulation after ligand binding. So they have a lot of functions related to their sites of origin. This is what happens in the amino acid when heated, and the production of the browning, which is a very delicious product in meat and chicken when they are exposed to heat. And these are some of the products that usually present in regular foods, which we usually eat every day, adding to the age in our body that's not only produced endogenously, but can be increased by eating the advanced glycation products containing foods. And the age receptors tails, when activated, they weave a theme of vascular perturbations and inflammations linked to the pathogenesis of diabetes itself and its complication. And this means that the activation of the receptors is not only present in diabetics, or the presence of diabetes is a conditioned situation that can activate these processes, but also other situations other than diabetes can activate these receptors, and it may cause diabetes itself. However, this receptor was shown to modulate immune responses and inflammatory processes in many conditions, including diabetes, of course. And this supports the hypothesis of the inflammatory region that is responsible for the development of diabetes and complications, whether it is in the macro or in the microvascular vasculature. However, let's go deeper into the receptors and its action. As we have said before, that they are an immunoglobulin superfamily members, as you can see from the slide, and it has a lot of ligands, as you can see here. And astonishingly, that the oxidized LDL is one of these receptors or these ligands, and if it is attached to these receptors, they will induce the same sequence of events that's produced when increased glucose, and the products are attached to the same receptor. So even the hyperlipidemia with increased production of oxidized LDL when present can lead to activation of the receptor without diabetes. If you look again to this list here, we can find that it can be activated again by the islet cell amyloid polypeptide, which is a product of hyperinsulinemia resulting from increased stimulation of the beta cell. We can find here again prothrombin. We can find lipopolysaccharides that can be product of many infections. So the attachment of any of these ligands, other than the age routines, can induce the sequence of events leading to increased inflammatory processes, increased fibrotic processes, and increased proliferation of the smooth muscle cells and other in other organs. Again, you can see here that the scissor-like movement of these receptors induces some sequences leading to increased reactive oxygen species production of nuclear factor kappa beta, MAP kinase, JNK, and PERK. And these will all lead to the complications that have been just mentioned. However, you can again see another way of stimulating or activating the receptor by transactivation, where a neighboring receptor can activate this receptor, RAGE receptor, through transactivation. And these have been shown by angiotensin receptor 81 and MCP1, interleukin 8 and interleukin 1 beta. These can transactivate the receptor. Here is an example of an 81 receptor transactivation to the RAGE here, and this will induce the same sequence of events that are produced when advanced glucose glycogen end products attached to the RAGE. And here is the effect of this transactivation on the plaque area in the aortic arch here. You can see here that in diabetic, the aortic area, plaque area is much more than those who have been wild animals with ApoE knockout plus diabetes. However, when we block the age receptor here, you can see how much is the reduction of the aortic arch plaque area in these animals. And that's the danger associated molecular pattern of these receptors and its ligands leads to the release of reactive oxygen species, nuclear factor kappa beta, BKC, RAS, and TGF beta, and this will all lead to the complications of diabetes or the development of diabetes itself. Let's look into what the literature tells us about the RAGE in proof of its role in diabetic complications. Here is an article published in 2014 in Transendocrinology and Metabolism, looking into the unblocking of the biology of RAGE in diabetic microvascular complications. And in this slide, you can see these are comparison between diabetic patients and the control when there is an overexpression of the RAGE receptors. The effect on the control is somewhat pathologic changes in the glomerular pattern here after 4 months of overexpression of the RAGE receptors. However, in diabetics, this effect is much more exceeding that of the happens in control. And this means that the role of the RAGE receptor here in diabetics is much more relevant to diabetic complications than it does in control. However, if we look at what happens in the if we will block the ACE receptors in both control and in diabetic patients, and here is the control glomerulus when the knockout ACE rages are there while still using the AT2 as a transactivation, and you can see that the deposition of the matrix collagen is much less in diabetics when we block the ACE receptors here. This happens in the retina as well and happens in the aorta as well here. And as you can see, the impact of blocking the ACE receptors is much more observed and it's robust than blocking the ApoE receptor, as you can see here. So the role of the ACE receptor here is much more relevant to the complication than the hyperlipidemic receptor ApoE is thus. Again, this is the angiotensin 2, addition to ApoE knockout aortic areas, plaques, and here is the effect after blocking the ACE receptors. As you can see here, the effect is non-comparable between the two. Again, here is the oxidative stress markers in the left ventricle of the mice, and it was shown that when we block the ACE receptors, the production of the oxidative stress superoxide marker here is much less than in the non-knockout mice. Again, this is another proof that the angiotensin 2 induced hydrogen peroxide as a marker of oxidative stress in animals that have been blocked, knockout of the RAGE receptors is much less than those who didn't be knocked out. This is again the depletion of RAGE, the receptors in the bone marrow, where it stimulates the production of the inflammatory cells that will start and initiate the atherosclerotic process is much more inhibited by the knockout of the RAGE receptors in the bone marrow, as you can see from this slide. However, one exclusion of this is the neuropathy, where the blocking of the RAGE was not found to have any relation to the severity or progression of neuropathy in diabetic patients. Let's look about the important thing, what happens when we block the ACE receptors in reality. Are there any proof from the literature that blocking these receptors can lead to any benefits? Here is an article that have been published 12 years ago, where the use of a substance called soluble RAGE can block the ACE receptors. And here's a trial that been done to show if there is any effect of this substance to attenuate the development of atherosclerosis in diabetic patients. And the study provides an evidence that the RAGE in reality have playing a central role in the development of accelerated atherosclerosis, which is associated and hallmark of diabetes. And these findings emphasize the potential utility of strategic targeting these receptors in the prevention of treatment and treatment of diabetic macrovascular complications. Here is a slide showing you that the knockout or the blocking of these receptors, the RAGE receptors with the soluble receptor, soluble RAGE, can alter the expression of the diabetic plaques, marker of atherosclerosis in diabetic patients. As you can see here, the number and the extent of the plaques are much more diminished here in those animals where the rage have been knocked out. Again, this is the suppression of the new intimal hyperplasia induced by balloon injury in the carotid arteries after knocking the rage receptors by the soluble rage if compared with the control and diabetic patient. You can see how much the new intimal hyperplasia have been reduced. Again, here is the kidney where the induced renal injury is much more reduced or by using the soluble rage as a blocking agent for the rage. Again, here is the expression of the nuclear factor kappa beta when stimulated by the 81 receptor is much more attenuated by the use of soluble rage as an approach to blocking the rage receptor, leading to decreased inflammatory process, decreased fibrosis, decreased proliferation of the matrix. Again, here is another proof that the effect of rage transactivation and inhibition through blocking this 81 receptor activation of the rage can induce much more less and less arc plaque area in diabetic patient with rage blocking. This happens also in the kidney, where you can see that the album creatinine ratio, which is a marker of the kidney function, has been much less affected by the inhibition of the rage receptor by the soluble rage substance. However, there is also a lot of research that have been done and published in the literature, and in this paper published in Clinical Diabetes Research 2017, 3 years ago, that shows that the review of the literature shows that we still have a therapeutic agents that can target the age, rage, axis as an approach to treatment of diabetes and its complications. And this has been done in the Mansoura University and in collaboration with the Monash University, Malaysia. And again, here you can see that in this paper published 3 years ago, we have a lot of agents that have been discovered to block the advancing glycation in products. And this approach is a novel therapeutic strategy, new strategy for treating diabetic vascular complications. And here you can see a list of some of these drugs that can be used in this way. However, here again, you can see that the approaches to target the pathway of the age and age receptor, the blockade, have succeeded to have an effect in the animal studies to reduce the nephropathy and atherogenesis in these animals and in humans. To conclude that we have said that the paradigm of rage have been changing, and now we have new knowledge about the rage, and we can see that it's more than one receptor. There's multiple receptors that are under the name of rage 1, rage 2, rage 3, and other receptors that can accept the ligands, and this all leads to development of diabetes itself or its complications. We have extensive data in animal models and human subjects, place the multifaceted families of these receptors and its ligands in diabetic tissues as key promoters of diabetes and its complications. Again, we have compelling data in animals using soluble rage and other inhibitors or rage deletion underscore that the relationship is not solely that of a biomarker, but likely of a mechanism. The current studies and those in progress aim to discern the precise expression patterns and potential functions of rages in diabetic complications. And if such studies are successful, then an entirely new class of intercellular based rage signaling antagonist may be on the horizon. Time will tell. Stay tuned. I would like to thank you for your presence with me in this talk, and I hope that I have added anything to your knowledge. Thank you so much for being with me here.